Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension Wisconsin 4-H, and on behalf of those folks and our other co-organizers, the Wisconsin Alumni Association, PBS Wisconsin, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. This week is Brain Awareness Week, and I'm delighted that Anna Garch was able to line up our speaker today, Jerry Yin, who's with both the Department of Genetics and Neurology here in the School of Medicine and Public Health. He's going to talk with us about what can fruit flies teach us about the persistence of memory. I'm now going to ask Jerry the 5 questions. Jerry, where were you born?
Denver, Colorado.
And where'd you go to high school?
Buffalo, New York. Suburb of Buffalo, New York.
Lackawanna? Lackawanna?
Amherst.
Amherst.
Lackawanna.
Lackawanna. I'm not going to make the Lackawanna joke. Okay. And then where'd you go for your undergrad and what did you study?
I went to Princeton. I was East Asian Studies and Religion major.
And then where'd you go for your advanced degrees and what did you study?
I got my PhD here in molecular biology.
How do you go from East Asian and Religious Studies to a PhD in molecular biology?
Well, that's a long story, but I would say it actually helps thinking about things. So we can talk about that over beer.
We can talk a lot about, yes. Okay. And then did you do any postdocs or did you?
Yes. After I graduated from here, actually I kind of had a roundabout way. I went to China for a year, worked with a joint venture that Promega sponsored there in a small city called Loyang. And then I came back and went to MIT for a postdoc, did a second postdoc at Cold Spring Harbor Lab. I was promoted there, and I moved here in 2004. 2004.
Well, thank you very much. I'm looking forward to this. It's a great way to mark Brain Awareness Week. And would you all please join me in welcoming Jerry Yin to Wednesday Night at the Lab.
Thank you. I should say at the outset, it's great to see real people in an audience. So we all instinctively know what memories are. And they come in a lot of different categories. You can ride a bike if you haven't been on a bike for a long time because you remember how to do that from school or college or graduate student era. You learn and remember autobiographical events, historical events. You might remember mathematical formulas. You might be able to hum and sing the sounds of silence. So those are all really different kinds of memory. And the question is, how is it that we can store those for such a long period of time? So the talk today is going to be, I'm going to spend about maybe half of it introducing memory formation and what we know about it. And then the last part, maybe it'll be 2/3, and then the last 1/3 will be about the work in Drosophila, telling us something, suggesting very strongly some things about how persistence occurs. So by now, this is the modern version of phrenology. We know the brain is got functional anatomical regions shown over on the right, and these anatomical regions are involved in all sorts of motor, cognitive, language, et cetera, et cetera. We also know that between the regions, that neurons and astrocytes are the cell types that are involved in connecting the different regions. And obviously, the regions have to be connected properly in order to function. Just as a daunting sort of couple facts, the human brain has around 10 to the 11th neurons, and it's estimated that it's got somewhere between 10 to the 14th and 10 to the 15th synapses, which are the points of connection between neurons and neurons. So that's a pretty large set of numbers. And the real question historically was, how do you approach studying a problem that's so complex? Like, how do you do that? And the historical pattern is that you try to reduce the complexity of the problem down to something that you can bite off and chew on. So some experimentally tractable set of experiments and observations and data. So there are going to be 4 historical threads that come together here. First is Ivan Pavlov, who, as you know and remember from introductory psychology, was really the first person that experimentally, or in a systematic sort of way, dealt with how animals learn to associate a neutral stimulus, a whistle, with either positive or negative reinforcement, in this case food, the positive one, and have it be that the whistle comes to represent the imminent presentation of food. And this classical conditioning, Pavlovian conditioning, is largely thought to underlie most, if not all, more complex memories and learning, okay? So from Sounds of Silence to cerebellar things, there's going to be principles that whoops, this is advancing, that were learned that apply. The second thread comes from the work of Eric Kandel, who won a Nobel Prize in year 2000. And Kandel reduced the problem to synapses, okay? So that's his major contribution for which he got the Nobel Prize. He's contributed other things as well. So what he did was he took a really simple organism, this sea snail, Aplysia, and he taught it a simple behavior, sensitization. Sensitization is the fact that when you hear an unexpected sound or experience something like that, you startle and then you pay more attention after that presentation of that sound. So he took this Aplysia and he delivered an electric shock to its tail, and then that's the sensitizing stimulus, and then gave a light touch to the siphon, and that led to a modification of a reflex, which was gill withdrawal. So it's a defensive thing that the animal does. And they worked out the details of the behavior, and then they worked out the circuitry, and that most of the circuitry is presented up here, and focused all of their work, almost all of their work, on the synapse, the connection point between the sensory neuron that comes from the stimulus and onto a motor neuron that controls the muscle that leads to gill withdrawal. And this synapse turned out to be, its efficiency was modifiable. And it turned out that there were other neurons that synapsed onto the presynaptic part of that synapse, the sensory neuron, and those neurons were modulatory neurons, and they came from the electric shock, the sensitizing stimulus, okay? And so that efficiency of transmission across the sensory to motor synapse is the one that Kandel's lab focused on. So they reduced this whole problem down to one synapse. And, in reality, that one synapse only represents a fraction of the change in the whole animal, but it represents a significant part. And then they went on to look at what was happening inside the sensory neuron. And they characterized a small molecule, a metabolite, cyclic AMP, and a downstream effector. So when levels of cyclic AMP in cells change, this protein called protein kinase A, or PKA, gets activated. And that it then phosphorylates or modifies other proteins, and those proteins change the efficiency of transmission. So it's more efficient, all right? So that's the second thread, a reduction to synapse, all right? The third thread would be a reduction to molecules. So Seymour Benzer, totally independently, was a physicist who started off in physics and then lost interest and was part of that wave of physicists that moved to biology in the late 40s and early 50s. And he got interested in the sort of core problems of molecular biology, molecular genetics, i.e., how does DNA make RNA, make protein? And he focused on a bacterial virus, a bacteriophage, that lives and kills bacteria. So what is shown over on the right is a part of a petri dish. And on the surface of the petri dish, there are bacteria. It's a lawn of bacteria. And the holes that you see are the plaques where the virus has entered many bacteria and killed them. And the clear area represents the kill zone. Those are dead, no bacteria in those zones. So what Benzer did was his lab focused on, because he came from physics, right? So this is really important. He came from physics, so his mentality was, I want to look at the fundamental unit, because he had come from atomic physics, right? So he was interested in what affects the way that plaque looks. And you see there are a couple plaques that are much bigger than most of them. So what can you mutagenize? What can you change on the virus that will result in a bigger plaque? And so in genetics, that is kind of like a genotype-phenotype relationship. That's how we would call it now. So he worked on that, and he discovered this R2 gene, and he discovered that what is most important is that you can mutagenize or change the single nucleotides in the part of the gene that codes for protein, and that those changes, some of those changes, result in those bigger plaques. So when he lost interest in this and decided he was going to move into behavior, he had to pick a system and pick an approach. And what Benzer did was he did the reduction to molecules and genes because that had been so successful in elucidating how DNA makes RNA, makes protein. So he picked Drosophila, the fruit fly, and he said, without knowing anything about brain anatomy, circuits, anything, synapses, nothing, he said, I'm just going to take the fruit fly, I'm going to randomly mutagenize it, randomly change genes within the fruit fly, and assay for those changes that affect learning and memory. And that's what his lab did. And shown on the left-hand side is a so-called memory decay curve. So this is, on the y-axis, is performance of the flies as a function of time, on the x-axis, time after the end of training. And the top line, which says control, are unmutagenized flies. So these are wild-type flies, and you can see that if you test them at t equals 0, immediately after training, close to 100%. And this down here is how they calculate it, but it's basically a simple algebraic representation of the fraction of flies that picked the correct odor. So the training paradigm they had was one odor is paired with electric shock, foot shock, then a second odor is given, not paired with anything, and then at some point in time, the flies are forced to choose between odor that was paired and the odor that was unpaired. And obviously, if the fly learns the association between electric shock and odor number 1, it picks odor number 2. So what you see is that if you test wild-type flies right away, they perform very well. But over time, as the interval between the end of training and the time of testing increases, the performance decreases. And that's forgetting, right? The two mutants, right, remember their approach was, okay, I don't care about brain regions, I don't care about circuits and synapses. Their approach was to isolate mutants that perform poorly. And the first two mutants that they isolated had the colorful names rutabaga and dunce, and you can see that they don't learn as well if tested immediately after training, and that their memory decay curve is different. The miracle of this was that when they then went to characterize what those two mutations, what the wild-type gene in which the mutation resides, when they went to characterize that, they found that one was involved in making cyclic AMP, adenyl cyclase, and the other was involved in degrading cyclic AMP. So totally independently, in a value-free way, they had come to the same molecule that Kandel and his workers had come to in aplesia. So reduction to molecules and genes. And then the fourth approach is the last one that was picked up, and this is more a traditional biochemistry sort of neurophysiology approach in which people cut out a slice of the hippocampus. So the hippocampus is a brain region that was well known to be important for memory formation in higher vertebrates. So they took rats, cut this section of the hippocampus out from the rats, and they did electrophysiology, okay? And this part of the brain has a very stereotyped wiring system. So signals come in from the dentate gyrase, they go to this region called CA3, all these neurons in CA3 then innervate the region called CA1. So the neurons in CA1, you can think about them as vertically, their receiving ends being vertically displayed, and all these different CA3 neurons come in at different levels, okay? Now, why was this such a important choice? Well, so this represents CA1 and two inputs at different vertical levels, okay? And those inputs are coming from different neurons. So what was discovered was that you could deliver, so the paradigm here is you first do a so-called test pulse, and then you see what the response in the CA1 neuron is to a test pulse. And you do a number of test pulses, so you establish a so-called baseline. This is what the average response to the test pulse is, all right? So the first experiment, you do a stimulus. It's not a super strong stimulus, it's a weak stimulus, and you ask, does that change the baseline response, the baseline reaction to a test pulse? And you can see there's a very transient, and so the more vertical domain is increased responsiveness on the postsynaptic side, the receiving neuron. And you can see there's a slight increase, but then it goes away very quickly, okay? So subsequent test pulses are really back down to before you delivered the weak stimulus, okay? However, if you do a really strong stimulus, right, then you see, that's number 2, what you see is that there's clearly, in response, the subsequent test pulses produce a much stronger effect on the postsynaptic side. But the key observation by Tim Bliss and Terz-Lomo was to pair the weak and the strong within like milliseconds of each other. And when you do that, both the weaker input, it shows a strong potentiation as well as the strong input, which normally produces a strong potentiation. So this was the feature. There's an association between the weak and the strong onto this neuron, right, same neuron. And of course, the reason that that was so exciting because this was a hint and provided a very strong suggestion about how Pavlov's dogs associate these whistle and the smell of food. That somewhere in the brain, in the dogs, those two sensory stimuli will come together, and perhaps the strength of the food reward is the strong stimulus, and the whistle ordinarily being a weaker one, but they become associated. Okay, and so subsequently, whistle will signal to the dog, and at those synapses, it will be treated as a strong stimulus. So that's one reason this model became so popular. The second is we already knew the hippocampus was important for memory formation. This thing develops rapidly. This is milliseconds, okay? And it lasts a long time. So this is a cartoon, and this is not important. The actual molecules and things, names are not important. But the idea, the important point is that experimenters treated this LTP, and they started researching and getting results consistent with an early LTP. So you do the electrophysiological treatment, and you get a response right away. And that increase in synaptic efficiency between CA3 and CA1 neurons, that turned out to involve lots of molecules in the pre- and postsynaptic, and this is a cartoon of the postsynaptic receiving end. But what I want to draw your attention to are two of the molecules, adenyl cyclase and the downstream effector PKA, which had come out of the Kandel and Benzer work on invertebrates. There are other molecules. Okay, I'm not saying these are sufficient. I'm just saying these are important. CAM kinase, et cetera. So the idea is that the short-lived increase in efficiency involves these kind of proteins, kinases, that get activated, and then they modify proteins that are down in this region. And those modifications, represented by the P's up there, lead to increased efficiency of reception of the signal from the presynaptic side. Okay, so this is like a general description of a whole set of mechanisms to increase synaptic transmission, the efficiency. After a number of years, because of the ascent of molecular biology, and the realization that DNA makes RNA makes protein, and the fact that we had drugs, pharmacological agents that could block the transcription, the DNA to RNA, or translation, which is the mRNA to protein step, people started asking, well, what happens if you apply these pharmacological agents to the LTP? So what is shown here, the take-home message here, is that these inhibitors of macromolecular synthesis do not really affect early LTP. So you add the inhibitor and early LTP shown in the, so this is the time when the drug is present. These are the test pulses. Now we've done the high frequency, we've done some strong set of stimulation. You've induced LTP. In the presence of inhibitors that block transcription or inhibitors that block translation, they don't really have a big effect on early LTP, right? So his potentiation being from 100 to 200. But what you see is that as you wait longer and longer, the curves peel apart from each other. And so those experiments where inhibitors are added, the potentiation decreases much faster. So the idea was you need, the implication was you need macromolecular synthesis for late LTP, for the persistence of LTP, but you don't need it for early LTP. Okay? So, and this is just a more recent rendition of this model, which is, turns out to be universal as far as we can tell. And the important thing here is just the emphasis that the fact that you need to recruit gene expression because you need to make new RNA and you need to make new proteins means that you have to send a signal back from the synapse to the nucleus, where that's the place where all the mRNA synthesis occurs, and then RNA synthesis occurs, and then some of that RNA is translated into protein, and some of the RNA in the cell body, and then some of the RNA may be transported out into the processes near the synapses and not translated until they get out there. Okay, so that's our modern version of these stages and kind of a cell biology cartoon to layer on top of it. So, okay, LTP, you got early LTP, you got late LTP. One requires proteins get modified, one requires new gene expression, new proteins to be made. It's generally accepted by researchers that it's unlikely there are too many totally novel new molecules that we don't know about that are going to contribute to persistence. Remember, our main question here is how do memories last? What I've described really only explains how memories last for a few hours, perhaps out to a day, certainly not decades. So that is really the question then. If all the mechanisms and most of the players that we know are really sort of, they get activated, molecules get activated, and they stay activated for minutes, they do something, gene expression, takes maybe 30 minutes, maybe an hour, and its effects maybe last for hours. How does persistence occur? Because you don't retrain every day for decades, right? And what is this activity, right? I emphasized that proteins like kinases get activated. They need neuronal activity to become active. It isn't like the protein is just made and it's active. The protein is made and kept inactive. So its activation requires neuronal activity. So what is the neuronal activity a couple years after the original memory experience? So those are the kinds of issues that the general question that we're thinking about involve. Okay, so now I'm going to explain what we do in flies, and then we'll talk about our recent data and how it contributes, I think, very strongly to suggestions about answers to these questions. So this is a picture of the apparatus I described briefly before. Flies are in this upper tube. The tube is, the flies are kept in the tube. They can't escape. There's a wire mesh at both ends. There's an electrifiable grid on the inside perimeter circumference of the tube. And so they're just walking along on the grid. The tube is hooked up to a moving air stream, and so we can bring an odor through the tube, and during the first minute, or step number 2, when the odor is brought through the tube, we deliver pulses of electric shock to the feet of the flies. Air is used to clear out the odor, and then a second odor is brought in without any pairing to electric shock. So then air clears that one. So at some subsequent point in time, the flies are placed into a T-maze choice point. So they're dumped right there, and they're simultaneously exposed to one odor that comes in one arm and exits perpendicular into the screen, and the other odor comes in the other arm and exits perpendicular into the screen. So they're placed at a choice point. They're simultaneously able to smell both odors, and of course, if they learn the association, they avoid odor A in this particular cartoon. So this is the memory decay curve again, and now you notice that we've changed our units to days, not minutes. So t equals 0, again, is the end of training. And the red curve, which doesn't show up real well, which is the one that says 1x, that's the memory decay curve if you give the flies a single 2-minute, 2.5-minute training trial. In order, and so you can see that even by 24 hours after the end of training, the fly's performance is getting close to the starting point. In order to produce longer lasting memory, there are, just like all organisms, you give them repetition, so multiple training trials. And those training trials can come in 2 versions. One is you give them 10 trials right in a row. So each vertical line over here represents 2.5-minute training trial, and you just do one right after another. It takes about 30 minutes to do, when all is said and done, to do 10. And you generate the curve that's in the middle, the so-called MAST. So this cramming or mass training does, in fact, extend memory out to 4 days. But again, by then, it's back down to starting levels, right? In order to get them to remember beyond 4 days, you take the same 10 training trials, but you put a 15-minute intertrial interval in. So instead of cramming, you distribute the training, and this produces the top curve, the one that says spaced or distributed, all right? So everything I'm going to talk about is the spaced or distributed training. So it's just, so this is a universal property. This is obviously, you recognize that we do better with spaced training than cramming, but in fact, cramming can get you past that test the next day. May not get you to the end of the semester, but it gets you past the test. So this is a universal property. So now I'm just going to summarize work in the field from a lot of different labs. So it turns out we give these flies this 10-space training paradigm. It takes about 2 hours and 40 minutes, all right? But it turns out that it takes somewhere 5-6 days for the fly to fully consolidate that 2.5 hours experience. So that's the first thing, all right? That's a fairly long period of time in the life of a fly. These laboratory flies live maybe 6-7 weeks, okay? Flies also have a property that mammals have, which is called systems consolidation, and that's just a fancy way of saying the regions of the brain that initially are recruited to learn and form the memory often are not the regions where the memory ultimately is stored. So there seems to be some kind of, and I'll use the word transfer, but I'm using it in parentheses, some kind of movement or transfer from where it starts to where it ultimately resides. That process in mammals, for hippocampal-dependent memories, in rodents, takes 2 to 4 weeks. So instead of taking 5 or 6 days, it takes 2 to 4 weeks. As I have said, the fly memory lasts at least 7 days. And so when you compare the processes in flies with the processes in mammals, we see that the fundamental properties, systems consolidation, this kind of sequential processing, the fundamental properties look very, very similar, but it's just compressed. It's much faster in flies. And that's one of our advantages from an experimental point of view. Okay, so the first clue that we had about what might contribute to making memories last, all right, was we were involved, I mean, the only thing I'm known for is work on a transcription factor that is important during the initial stages of forming memory. And the transcription factor is one in all cells, all organisms, that responds to cyclic AMP, that molecule that was originally described by Kandel and Benzer, right? And when levels of cyclic AMP are altered, this transcription factor is activated or repressed. So we showed, and then it was shown in all animals, or all through rodents, up through rats and mice, that the activity of this transcription factor was critical for forming memory. So we made what is called a reporter for that transcription factor. So we wanted to know where in the fly brain the transcription factor was active, because it seemed to be necessary for making long-term memory. So we put binding sites for this transcription factor, because that's what transcription factors do. They bind to particular sequences, called CRE sequences in this case, in front of a simple promoter, and we used as a reporter, like a proxy measure for activation, we used the luciferase gene. So we took that cassette and made transgenic flies that contained that thing and asked where and when, what do we see, right? And of course the intention was to ask, what do we see differently after training the flies? Well, the first surprise was just, what do we see without training? And what is shown down here is time And this is 12 hours of darkness, 12 hours of light, so this is a week, roughly a week's time. Light productivity, the amount of light that can be detected from outside the intact behaving fly, so we're not cutting sections, we're not making extracts, we're not doing any of that. We're having flies in a little microtiter dish well, and there's enough light produced that a luminometer can detect it. The luminometer reads the flies once every hour for days and days, right? So what you see is that the activity oscillates, okay? So I was real surprised. I would just add that at the time we started working, there were about 30 publications on this transcription factor. There are now 13,000. Out of the 13,000, there's still only 20 that talk about oscillating activity. So this is what we see. And we know the activity, so you can see this is a 24-hour period, right? So there's one peak in the nighttime and one peak in the daytime. This activity is, truly what we think it represents, because if you take away the transcription factor by mutation, you knock the whole thing down. If you alter the best binding sequence for the transcription factor, you knock everything down. So it is reporting CREB activity as we hope for. Those of you with really good eyes might see that there's a switch from light-dark to gray-dark. And gray-dark represents constant darkness. So in the 12-hour light period, we take away the light. And that still results in oscillating activity. It changes the character of it a little bit, but that is the behavioral definition of something that's under the control of the circadian system. So when it's an oscillating phenomenon is established under light-dark, you turn the lights off and it persists. That's the behavioral definition of something under oscillatory circadian control. The genetic demonstration is fancier. So in flies, there are mutants that are, my screw just fell off. There are mutants that affect the frequency of the clock. So you can take this reporter and cross it into these different mutant backgrounds. And the one in blue is where there's no clock. And you see there's no oscillation. The one in red is when you cross it into a fly that has a mutant period gene that results in a 28-hour period instead of a 24-hour period. And you can see that the frequency of the reporter changes appropriately. And the one in yellow is when you cross it into a mutant background that has an 18-hour period instead of a 24-hour period. And you see the frequency oscillation changes. So this is the genetic data. This is telling us absolutely this thing's under the control of the circadian system. Okay? So when we started thinking about what might contribute to persistence of memory, and this figured prominently. So the first positive experiment we had is the following, and I'll walk you through this diagram. It's a little complicated. The vertical striped box represents training. We train during the nighttime, so you see we have our blocks of 12 hours of dark and 12 hours of light, and I've labeled some of them. So the experiment is basically you train the fly, and then after training, we're going to mess around with light darks. The control flies get light dark, and then we test them for memory at the points in time represented by the black arrows. And the horizontally striped bars indicate darkness. So we're imposing darkness on what would have been a light period. And what you see right away, or what you see if you study this, is that if we train, and then the top line, if we train and impose darkness right after training and test the flies for 24-hour memory, there's really no difference between whether those flies experienced a period of imposed darkness or not. No effect. We're always comparing it to the wild type condition. However, if we impose dark in the equivalent of day 2, day 3, just day 2, or just day 3, we disrupt 3-day memory. So that telling us that something about light dark is needed for 3-day memory, but nothing about light dark affects 1-day memory. Okay? We then went in and manipulated molecules that are part of the circadian system, and we get the same result when we manipulate the molecules essentially beginning around N1, the middle of N1, out to 3 days. So there's a window of time where darkness, constant darkness, or manipulating, disrupting, interfering with the core circadian machinery disrupts 3-day memory. All right? This is a summary slide, and this is close to the last slide. So that got us into thinking, okay, maybe other oscillatory processes are also involved. So ecdysone, shown here in green, is the fly steroid hormone that's made in adults. And so it turns out, and I won't go through all the experiments, I'm just summarizing the experiments here. Basically, if you mess up ecdysone signaling, you affect 1-day memory, but if you mess it up later on, you have very small, negligible effects on subsequent memory. The circadian thing, shown here with the red, you can't see the red real well, that, of course, as I've emphasized, occurs, the sensitive period is between 1 and 3 days after training. The third oscillatory process that we know is important, I'm going to call growth control, okay? And growth control involves genes that are mostly associated with the cell cycle, okay? So when cells, you know those pictures of cells dividing and the DNA being pulled apart, that's all timed and regulated by the cell cycle. Now, it's important to keep in mind that these cells have exited the cell cycle. These are post-mitotic cells. They're not going through division, cell division and stuff. But nonetheless, at least one of the key regulators, the oncogene MYC, its manipulation of MYC, again, has a specific effect between 1 and 3 days, okay? I don't really know what's back here. I mean, I have some ideas, but I'm not going to talk about them today. So, but the important thing, the key point already, right, is all three of these, ecdysone, circadian, and growth control, are normally oscillatory phenomenon. And oscillatory phenomenon are important. So there are two sides to the coin I mentioned initially. One is how do memories last a long time? And the other one is why does it take such a long time to form memories, right? Flies take 5 to 6 days. mammals take 2 to 4 weeks for hippocampal-dependent memories. So if you're recruiting oscillatory processes, there's a requisite amount of time you need to synchronize them. In other words, if I'm taking a fly and I've got it waking up at 9 a.m. in the morning when the lights come on and going to bed at 9 p.m. when the lights go off, if I want to shift it from that schedule to getting up, waking up at 6 p.m. and going to bed at 6 a.m., that takes between 2 and 3 days. So the fact that we've got multiple oscillatory phenomenon that are required suggests that part of the answer for why it takes a long time is that you've got to synchronize and align these things, okay? So that's one point. The second point is that it is now known that especially the circadian system, these oscillatory processes stimulate gene expression, and they stimulate the signaling pathways that activate the proteins that are made. So this is the likely source, or a likely source, of replenishing the molecules that contribute to persistence of memory, LTP, all that stuff, and to reactivating them on a periodic daily basis. And that, of course, is the third point, or down here, which is that oscillatory processes last forever. So maybe there's a replenishment and reactivation that occurs basically on a daily basis that contributes to the molecules that contribute to the persistence of things like LTP. Also, the fact that these processes occur every single day, they may provide windows, I'll call them susceptibility windows, times when the system is a little bit fragile and maybe new associations can occur, whether those are correct or true or false, okay? But it's just an idea. So those are the most important points about the data that we have so far. At least 3 oscillatory processes, and they go on forever. Now, you know, some of you are probably sitting there thinking, well, he hasn't said the S word yet, which is sleep. Well, you know, everybody knows sleep is important for memory formation. Sleep is regulated by the circadian system, okay? It's experimentally very difficult to really disentangle a direct sleep effect from a circadian effect. Those two, because the circadian system regulates sleep. So I would include sleep as a necessary recurring process that's included here. Okay, I'm just going to finish up. This is the next to last slide. This is a very interesting experiment that was done when the Challenger disaster occurred. Liz Phelps, who teaches psychology at NYU, happened to be giving a class on the day of the Challenger disaster. So when the students came to her class, she asked them to write down on a sheet of paper where they were the moment they heard about the Challenger disaster. So she collected those pieces of paper. She also kept in contact with the students, and I think it was 20 years later, she wrote to them and asked them to state where they were when they heard about the Challenger disaster. And a large fraction of the answers 20 years later did not match the answers that were given on the day of. So that's just a fact. It's a aspect of memory formation. Perhaps it's like, as I suggested a moment ago, when you have these windows of susceptibility, perhaps during those times, you're susceptible to enhancing the memory, augmenting the memory, maybe even changing the memory. And that might contribute to, the observation that Phelps made. So that's my talk. I just want to acknowledge all the experiments that I did, and of course I didn't present any of the data, but this is basically 3 years of experimentation, starting before COVID, and largely we were working through the COVID period because flies, you have to passage flies, you can't just let them sit, they die. The experiments are all done by Home, who's my wife, and myself. There are 2 undergraduates in our lab over the last couple of years. And these are people that have left the lab but have intellectually contributed to the thinking behind the work. So I'm going to stop. I'll be happy to take questions.
Questions from the audience? I'll start back here.
Hi, great talk. I have 2 questions. One's a geeky one. What about constitutive expression of CREB outside of when it should be expressed? But also the bigger question is it on? I think so. Okay. The bigger question is, the training that you did is repetitive, but the Challenger was a single time. I don't remember where I was during the Challenger explosion, but I remember it. So that wasn't trained. That was like...
Yeah.
So what are the differences there?
So I'll answer the second one first. It's likely that highly emotional events recruit the amygdala, which is the emotion center of the brain, simplistically. And so you will have a heavy input to the memory circuits from the amygdala. In fact, the memory circuits might be in the amygdala. So this is one of the things, neuromodulatory factors can basically substitute for repetition, I mean, at a molecular level, all right? That's how I would describe it. I think it's a fairly even-handed summary of the work in the literature. The first question, what happens with constitutive CREB and CREB that's expressed elsewhere? So there's 13,000 papers on CREB, and there's a reason there's 13,000 papers on CREB. That's a great question. I did not talk about a whole nother, so I emphasized memory and changing the efficiency of neurotransmission at the synapse. There is a second mechanism, at least a second mechanism involved, where the excitability of the whole neuron is altered and increased. So when you take an animal and train it, if you cut the hippocampus, you train it with a hippocampal-dependent task, and then right after training, you cut the hippocampus out, and about 25% of the neurons in the CA1 field are part of the circuits that represent that memory. You can find those neurons. They have increased excitability. And what that means is that not only what that means is that all of those neurons fire at a lower threshold. It takes less to fire them at all their synapses, not just the one that was part of the circuit, okay? And what CREB does mechanistically is change the excitability of neurons. But it is also recruited when something strong happens at one particular synapse. So there's 2 mechanisms that contribute to increasing synaptic efficiency. One of them is synaptically specific, One of them is more general.
Yeah, just two brief questions. First, could you describe what medium do you use, what media to create the different odors for your research? And then also, did I understand you to say that the fruit flies sometimes emit light? And if so, what causes them to emit that light? Thanks.
Okay, so the odors are presented on humidified air. Okay, so it's presented as a vapor, sort of. Okay, the odors are, we didn't work this out. The odors are picked so that without training, they have equal, they're mildly aversive. Okay, so if you give the fly a choice between one of the odors and air, without training, just boom, plop them in a teammate's choice point with odor on one side and air on the other, they prefer air. So they're mildly aversive. So we're taking, but they're balanced. They're equally mildly aversive. So we use concentration so that if you give the odor on one side and you don't train them, just give them on one side, give them on the other side, they split 50-50. Okay, so that's important, right? The light-producing flies are artificial. We have put a gene into them. We made a transgenic fly that makes luciferase. We supply the substrate for luciferase, luciferin, and luciferin takes ATP and produces light. So that's we're measuring how much luciferase is being expressed and using light as a proxy for the transcription, which is driving the luciferase.
Right, and that's the same process that occurs in fireflies?
Yes, this is the firefly luciferase gene. Yeah, yeah.
A question from online, from Raj. In the fruit fly experiment with odors, instead of a single fly experiment, had we used multiple flies, is there any impact on memory retention from a social behavior point of view?
Good question. We actually use multiple flies. So our experiments involve populations of flies, and that has the advantage that it gives us larger numbers and bigger n's and stronger statistics. When this paradigm was originally worked out, Tim Tully, who worked on it, actually did the experiment where he trained 100 flies, but one at a time, and looked at calculated their performance versus 100 flies trained and tested in groups. So there was no difference. So at least under those conditions, the social thing did not. Now, I should say, in my heart of hearts, I think there is an effect of social groups because flies are very likely, like all animals, to produce pheromones that are alarmones. So when they get electric shock, they don't like that, and it's likely that they're producing an alarmone, and so there is a more complex situation when multiple flies are there. But the point is we train everything in the same groups and test them, and et cetera. Every experiment we do here, an N of 1, all the experiments we do, we involve N's of 8, if you're a statistician, and an N of 1 is 200 flies. And it's 100 flies that are trained with odor number 1 paired with electric shock, and then 100 flies trained with odor number 2 paired with electric shock. So we're using balanced experimental approaches to bolster our strength of our statistics.
Thank you.
That was a good question.
Okay, Jerry, thank you and Hong for doing all these experiments during COVID. At least one thing that is good that happened during COVID.
It's better than doing nothing during COVID.
Right.
Yeah, I was gonna say.
Have there been any studies to compare the training length and the memory retention, or those windows of opportunities between adults and during development stages? In other words, we always hear that when you're younger, you are much quicker in learning, you are much, you remember longer and so on.
Yeah, that's a good question. No, there has not been a thorough, exhaustive study comparing what you might call teenage flies and what you might call old age flies like me. But the practice is we use young flies. So we use them. So flies come out and they, it's estimated it takes a couple days for their maturation, so adolescence, if you like. We typically train flies that are about a week old, or a few days to a week old. We just keep that constant as much as we can. But the deeper implication here, I didn't have time to talk about this, but those oscillatory processes are all players during development, okay? It's not like these just sprung out when adult flies emerge, right? So, pretty much everything that I think about now, I'm looking at development because that's where the evolution first evolved the solution. And this solution has something to do with timing. It has to do with multiple, I think, multiple oscillatory phenomena needing to be integrated and synchronized, and it is timing thing. So, the real evolution of timing was during development. Yeah.
From online, Diana says, very interesting, thank you. I'm now trying to figure out how circadian rhythms contributed to my success or failure to remember study material. Not a question?
Yeah, so
pretty cool comment.
So yeah, that's an interesting. So all animals show time of day dependence. That is, so for flies, if we take the 24 hours and divide it into 6 time points every 4 hours, and we train and test them, there is 1 out of the 6 time points which has maximum performance. There are 4 that have the same performance, and there's 1 that has really bad performance. And that's why we train at night, because it's near the optimal time. The underlying mechanism of that is totally unknown. And you'll notice we're training at night. We're not, so the intuitive thing that they might perform best during the daytime when they're awake is not what we see. All animals have time of day dependence. It's not well understood, well studied or well understood.
Okay. I have a couple of questions. This is a new idea to me, systems consolidation, and you mentioned the transfer of an original site to a hippocampus in 2 to 4 weeks in like mice. I'm going to refer to it as a copy of a memory. When you transfer that copy of a memory to the hippocampus, does a copy of that remain in the original place, or is there one copy of it and it gets moved and it's not left behind at the original site?
That's a controversy in the field, so that's a great question. So the memory starts in the hippocampus, goes to cortical cortex.
Sorry about that.
Different parts of the cortex.
I didn't remember correctly.
But the question is, And that's why I said I'm using parentheses around transfer. The question really is, does it really physically leave the hippocampus? Or is it that the brain is much less dependent and reliant upon the hippocampus and is using circuits in the cortex to drive the behavior? And that is very much up in the air. Most people do favor the idea that, well, experimentally, it's less dependent on the hippocampus, okay? But you can find people with data that says there's still some dependence on the hippocampus. And what that really means is, as a scientist, when you have conflict, this is my personal philosophy, when you have conflicting data and both sides have done the experiments correctly, there isn't like a technical problem, what that tells you is that it's variable. And that means that there's some parameter or parameters that determine how much 1 month memory depends on the hippocampus or not. And we have no idea. There's nothing known about that.
And then because it's Brain Awareness Week and there aren't a whole lot of songs from my youth that featured memory, there's that great song In the Year 2525.
Oh, yeah.
Yeah. And the line is, everything you did, think, or say was in the pill that you took today. Since this is going to be one of the latter questions, what do we have, not from your research, but what can you tell us on Brain Awareness Week for inducible memories, whether it's, well, I'll just go with the idea, are there ways to either induce genes, transfer genes, or take a pill to induce a memory?
Well, when the CREB work happened, there were 3 independent biotech companies that formed, and all of their research and business plans were around CREB, because CREB is conserved in humans as well. It wasn't really well appreciated at that time. The systems consolidation stuff had started coming out, but it wasn't widely appreciated. What I would say now is that CREB is likely to be involved in all of the steps during systems consolidation, but it isn't like you want to enhance CREB everywhere all the time. What you really want to do is only enhance it at specific places at defined times for, enhancing memory. And that's something that pharmacologically we don't know how to do yet, right? You can't take a pill and only have the compound work in visual cortex and whatever. Do I think it's possible? Well, I still think it's possible to pharmacologically enhance memory, but it's one of these things where I think we need more research. I'm going to sound like, whatever. We need more money, more research to enable to get us to that point. Yeah.
Any other questions? Before I go to you, I want to say to everybody, we're going to go next door to the library for memory-inducing beer. Memories. So if any of you'd like to join us, I got to close up shop and everything, but I'll buy, and we can go next door, and here you go.
Thanks. Yeah, this is a very unscientific question. You used the example of the Challenger disaster as a basis for people remembering what they were doing at that time. I'm wondering if you'd get a more higher incidence of people recording correctly if you used the 2 more classically used dates for that, specifically, the assassination of JFK or 9/11. Thanks.
Yeah. Well, again, I don't really have an answer to that other than I think emotional input is a big part of this. So perhaps Kennedy's assassination and certainly 9/11 had greater emotional impact on most people. The average impact on the average person was probably larger. That's what I would say. You know, having said that, it's not like all our memories are false, right? Because you can hum the sounds of silence, you can sing the words. There's nothing wrong with, except your voice, with the, you know, singing part. So, you know, it isn't everything. It's just that it is a feature of memory formation that some memories are maybe not quite totally correct. Right? Yeah.
All right, thank you again.
Okay, thanks.
So next week we'll have the talk on physical therapy of the hip and knee. And keep in mind, UW Science Expeditions, April 8th, 9th, and 10th, the 20th annual campus-wide science open house. See you next door at the library in a few minutes. Thank you.